Morning, everyone. It's great to have you join us this morning for this really important event in the Faculty of Science, Sun and Science. My name is Bruce Lennox. I'm the Dean of the Faculty of Science. And what we're doing today, some of you might be familiar with it, but to those of you who aren't, this is a showcase that the Faculty of Science has been featuring now for more than 11 years under the banner of Soup and Science every September and every January. But we've taken this special opportunity that these special conditions have provided us to actually do this in the summer for the first time. So this is our 13th this summer, Sun and Science. And the perspective that we take is we recognize that undergraduates, especially early undergraduates, if you're going to be a U0 or a U1 student this year, aren't very familiar with the concept of what research is. You're not very, that's our 11-pound dog that you hear in the background. You're not familiar with what research is. You hear about it. Your professors will be teaching your courses to you, but they also have a second part of their life, and that's as researchers. And that's what we're going to be talking about today. So what we're going to do, have four speakers: Professor Audrey Moores from chemistry, John White from physiology, Bastien Castagner from the Department of Pharmacology and Therapeutics, and McVean, who works through the McGill Chemistry Outreach Group. So once the presentations are complete, you'll each receive a message from the host to join a breakout room. When you're in the breakout room, you'll be in a smaller group and you can ask the presenter the questions, and then you can rotate through rooms. So I think I'll then hand the podium over to either my dog or better to Audrey Moores, who's going to talk to you about her green chemistry research. So over to you, Audrey.
Okay, so, sorry. So my name is Audrey Moores. I'm a professor in the Department of Chemistry, and I'm going to talk to you today about how we can take crustacean shell and turn them into new plastics. So do you know what this is? So you see something that is operated by the wind and waves and currents, so it's got to be in the sea. And what are these little things here floating around? These actually are plastics. So this thing is actually a giant robot that is operated in the ocean now to try to do something that's called the ocean cleanup. This is a giant robot that is 600 meters long, and that is trying to capture things like this that are floating around in the ocean. As you know, we have a major problem right now with plastics because we've failed to design them properly and to think about what happens to them when they go out in the wild without us controlling what happens there. So that's something that Canada is particularly concerned with because in Canada, we generate a lot of plastics, 4.6 million tons. This is numbers from 2016. But most of it ends up being landfilled or incinerated, and only 9% of it is recycled. Worst of this is that 1%, so this is equivalent to 46,000 tons of plastics every year, goes to the environment, and in particular to our rivers and oceans, where they do great damage, as you know. So my group has been looking into ways that we could potentially fix this problem, and one thing that we thought of was to use something that nature makes itself, which is a polymer called chitin. So we turn to animals that are able to produce their own polymers. Specifically, crustaceans or also insects are able to produce a polymer called chitin. I don't know if you've ever had lobster. I really love lobster, as you can see from my background. And when you eat it, you can see that the shell is very hard and actually has properties similar to the ones of plastics. So unfortunately, a lot of this gets wasted. We generate huge amounts, millions of tons yearly of seafood waste, and we do nothing with it. Well, we do a little bit of it with it. And what we do is we take the chitin, which is about a third of the content of this guy, and we turn it into another polymer called chitosan. And what chitosan is used for is for making sutures, for making things for water treatment, or for making antibacterial fibers. So people are really getting incredibly excited about this material. But there is a catch, there is a problem. In order to take the crustacean shell and turn them into the desired chitosan, you need a lot of chemistry and some not really good one. So specifically, if I mix here, if I show here the mix that you have in a crustacean shell of the chitin, but the other components, you need to go through 3 treatments using harsh chemicals to turn this into the desired chitosan. And I'm going to focus on the last one here, which is turning chitin into chitosan, which requires really, really harsh conditions, in particular, the use of corrosive solutions of caustic soda, dissolved in water, and extensive heating for several hours. So the limitation of this process is that it uses corrosive liquids and lots of energy. And this is to the point that people are now, there's only a small place in Canada where they're making chitosan, and many companies shut it down because of the downside of this thing. So our group has decided to look into this, and in particular found if there would be ways that we could make this process cleaner, and in particular make this process a little bit differently so that we retain most of the properties of our chitosan. So we devised a method where we could take chitin, which you can see here in a crystalline form, and by a series of treatments that are happening in completely dry form without the use of corrosive liquids, we can turn them into the desired chitosan with very high yields. And we can do this across the board with a lot of different sources of chitin, for instance, shrimp, lobster, crabs, but also fly larvae and beetle. And this is really exciting because, as you know, the future has us eating insects. So that means that we're going to generate a lot of chitin and potentially a lot of materials. We went on and looked into formulating this, so you can see that we can take this material and formulate it into something that we can mold and shape. So we're now turning this into making real plastics with it. So if you're interested in learning more, just ask me any question and I'll be happy to walk you to the future of bioplastics from crustacean shells.
Thank you, Professor Audrey Moores. We will now have Professor John White from the Department of Physiology giving his presentation. And I will give you controls in just a second.
Well, good morning, everyone. Thank you for the opportunity to speak to you about vitamin D and its relationship, if any, to the COVID-19 pandemic. There has been a lot of not hype, or at least interest in the scientific literature, and some of it has filtered down into the popular press about the potential of vitamin D supplementation to either prevent or treat COVID-19. So a very brief introduction into what is vitamin D. First of all, strictly speaking, it's not a vitamin. We can make it ourselves in our skin in the presence of UVB irradiation from the sun, which actually there is a conjugated double bond in the last precursor in cholesterol biosynthesis, which is present in every cell in the body. This is UV sensitive, and it is broken in the presence of UV light to produce vitamin D3. Vitamin D3 is not a biologically active molecule. It has to undergo 2 modifications to become active. It passes through the liver, where it is hydroxylated at the end of the cholesterol side chain to produce 25-hydroxy vitamin D3, which, if you have a vitamin D test, is what most of the time is going to be measured. 25-hydroxy vitamin D has a relatively long half-life in the circulation and is the major circulating metabolite. That, in several peripheral tissues under controlled conditions, is then 1-alpha hydroxylated to produce the active or hormonal form 1,25-dihydroxy vitamin D, which binds to the vitamin D receptor, which itself is a major regulator of gene transcription. So if vitamin D is available from the sun, why do we call it a vitamin? Well, there's a catch. The angle of the sun has to be above 45 degrees, and in Montreal, that only occurs for about 6 months of the year. And when you're at the beginning and the end of vitamin D winter, you really only have a limited period of time around noon where your skin will make vitamin D in the presence of UV light. And if you live in Scandinavian countries, you can see that vitamin D winter lasts almost all year. What is remarkable, however, is that vitamin D deficiency is really quite widespread worldwide. And one of the places to go to find rampant vitamin D deficiency, in fact, is in areas like Saudi Arabia and the Emirates, which are very sunny and very hot, but where people dress conservatively and they avoid the sun whenever possible. And in fact, 60 or 70% of the populations in these areas are in fact vitamin D deficient. So this is illustrated by this slide, which is a study of 25-hydroxy vitamin D levels in populations in the UK. And you can see that they vary seasonally. And really, the population is considered to be, by most people, vitamin D deficient almost all year long. So what is the relationship of vitamin D to the immune system? Well, we know that this last controlled step, the production of 1,25-dihydroxy vitamin D, occurs in several cell types in the immune system, and it is stimulated by several types of immune inputs that are turned on when the body detects an infection. And in fact, we now know, this is research over the last 15 years or so, that vitamin D stimulates the so-called innate arm of the immune system and boosts defense against several types of pathogens, including viruses. Laboratory experiments have shown that vitamin D has antiviral activity against common respiratory viruses, such as influenza, and viruses such as respiratory syncytial virus, etc. There is also good clinical evidence that vitamin D supplementation at the right times of the year reduces rates of seasonal influenza. In addition, vitamin D suppresses peripheral inflammatory responses of the immune system, and this may be relevant to inhibiting the so-called cytokine storm that can be so lethal in patients suffering from COVID-19. So what is the link between vitamin D and the SARS-CoV-2 virus, which is the etiological agent of COVID-19? At this stage, there is no direct evidence that vitamin D has antiviral activity against the SARS-CoV-2 virus, for the simple reason it's too early. Here at McGill, there are level 3 containment facilities that are being set up and are just getting underway to be able to handle the virus. You can imagine something that is transported through the air in aerosols is a dangerous virus to study. But these studies will be ongoing soon, and I'm sure we're not the only university in the world who is studying this. In addition, there are dozens of clinical trials that have been set up to test the effects of vitamin D supplementation on various aspects of COVID, either prevention or outcome, and they're recruiting patients. I looked up clinicaltrials.gov yesterday. There were 19 registered, and I know of several others, including one in Montreal and nearby areas. So I've got a supplemental slide that I can refer to in the breakout rooms, but if you have any questions, I will be happy to handle them in the breakouts. Thank you.
Thank you, Professor John White. Our next speaker is Ada McVean from the McGill Chemistry Outreach Group. And Ada, you have controls.
Perfect. Okay, so hi, I'm Ada McVean. I'm a graduate student in the DAMA lab, and I'm a science communicator with the McGill Office for Science and Society. But today, more importantly, I am a volunteer with the McGill Chemistry Outreach Group. So we're going to do really quick 5 minutes. Who are we? What do we do? Why do we do it? And how can you help support us in our mission to bring science to the masses? So put very simply, who are we? The Chemistry Outreach Group, we are a group of students and staff and alumni who work to foster a deeper understanding of chemistry through fun and interactive experiments. More specifically, for who are we? We are about 40 students. There's 36 of us right now, but who's counting? We're mostly, but not entirely, from the chemistry department. Right now, I'm pretty sure we have a neuroscience student volunteering with us, and we welcome anybody from any scientific discipline, or even from outside a scientific discipline if you have the experience necessary for lab work. We're all volunteers. Nobody is paid for their time, the many, many hours that they put into this work, and we're all very, very passionate about science communication. We're made up of mostly graduate students like myself, but we have a fair amount of staff and alumni who we just can't seem to get rid of once they graduate. They really like to come keep volunteering with us. We have not as many undergraduate students as we would like. We usually have a little bit more. I think this year has been a bit weird for recruiting people, but we really do like to encourage undergraduate students. I was an undergraduate when I started volunteering, and I'm still here 5 years later. So it's really fun and definitely worth sticking around if you're going to be at McGill for the long haul. What we do, simply put, is do demonstrations. So we do demonstrations of different science experiments for different students and other groups that want to learn about science. Some of the time students come to us, some of the time we go to the students, and some of the time we host big public events here at McGill. So I guess that is still just another version of them coming to us. We often try to pick a theme. We are a chemistry outreach group, but we would be lying if we didn't say that a lot of our science explanations cross over into biology, physics, psychology, almost any other type of science, especially once little kids start asking questions. You can really quickly jump from why does that piece of paper burn to why is the sun burning? So we often try to pick a theme. That way we can really unite, get across one common concept. So some examples of that are light and color, chemical reactions, what is a reaction, how do we know a reaction has taken place, or green chemistry. So how can we make chemistry more environmentally friendly by making plastics out of lobster shells, or how can chemistry help combat climate change? A good example of that is our Nuit Blanche from this year, which my slide isn't going, sorry. There we go. Our Nuit Blanche this year, which is our biggest show of the year, was themed on green chemistry. So both literally green things. You can see us blowing up a hydrogen balloon there that we've included some copper chemicals in order for it to turn green, but also some chemistry that's about the environment. So we looked at GFP proteins, we looked at how we can work towards environmental sustainability, and we also just, made some big green explosions because blowing things up is really fun, and we'd be lying if we said that wasn't a decent reason of why we volunteer. I don't have even, in 5 minutes, I can't even explain one demo that we do in full. So here is just some rapid fire, the things that we like to do that are really fun, and you'll have to come see us in person or online if you want to see more about it. So we like to do something called elephant toothpaste. This is a superconducting magnet track we made, very fun. We made a hydrophobic lab coat. So we coated it in hydrophobic substances so that water blew off, and then we let the audience squirt me with water guns. We love to play with fire. Specifically, this was lycopodium, which is a very flammable pollen. We made edible water balls out of algae, so like an edible water bottle almost. We make cabbage juice indicators out of things that you have in your own kitchen that'll indicate pHs, and we encourage people to make them at home. We make pumpkins vomit because that's really fun. We make fruit batteries, and of course, more fire. This is specifically nitrocellulose, or you might know it as gun cotton. So the why we do this, there's a lot of varied reasons why the 36 of us that are currently members might be trying to do this. Everybody's got their own past and their own goals, but there are 3 main tenets for why we think that this work is really important. We want to teach people. Of course we do. We think chemistry is awesome. We want to teach people some chemistry. Might be a certain reaction, a certain type of chemistry, or just a certain concept in science. We really try to tailor our presentations for the age groups that we're looking at. So we might go as far as to teach grade 12s about something very specific to their curriculum, or we might teach kindergarteners about the very general concept of what science is. We also want to entertain people. Obviously, if people have fun, they're more likely to come back for another demonstration. And if they have fun, they're also more likely to tell people what they've learned, remember what they've learned, and it's basically free advertisement for us because they'll tell their friends how cool that demonstration from the McGill Chemistry Outreach was. And then we also, as our third tenet, and this comes partially because we're funded from the McGill Office for Science and Society and partially just because it's a good idea, is fighting misinformation and pseudoscience. As you know, we have maybe crossed the age of information and are living in the age of misinformation now. So helping to dispel myths about chemistry and science, fight chemophobia, in general, just encourage faith in the scientific process and teach participants to recognize bad science when they see it. We're also lucky to be a very diverse group. There's 36 of us. A lot of us are queer. A lot of us are students of color. A lot of us are women. And so we really do want to encourage students that even if they don't look like the traditional scientist, there is a place for them in academia. We're funded mostly through the McGill Office for Science and Society, but we also get a lot of really lovely support from Nuit Blanche, and that's our biggest event each year. I'd really encourage you to come see it. I don't know if it'll be in person or online this year. I guess we'll see. And a lot of the photos were taken by Matteo Zamaria Photography. And please follow us on all the social medias, not just because I run them, although I am biased, so I think they're pretty cool. But if you're interested in volunteering, please send us an email. Or if you're interested in getting us out to, if you have kids who have schools who would like to have demonstrations like this, Girl Guide groups, immigrant groups, really anybody you think could benefit from learning a little bit of science, that's exactly where we want to go. So please follow us on social media and drop us an email if you're interested.
Great, thank you, Ada, for that wonderful presentation. Our last speaker is Professor Betsy Castagne from the Department of Pharmacology and Therapeutics. And I will get your slides set up in just a second. And Bastien, if you just double click on your screen, you should be able to access the controls for your presentation.
All right, so I'm stealing some of the punch. All right, so hi everyone. So today I will tell you about what we're trying to do in trying to understand the human gut microbiota. So as you probably know by now, there are roughly as many bacteria in and on your body as there are human cells. And most of these bacteria will reside in your large intestine, in your colon. And there are hundreds of different bacterial species in your gut. And their genomes of these bacteria combined, what we call the microbiome, encodes for roughly 100 times more genes than the human genome. So that's a huge diversity that can be quite important. And indeed, these bacteria are not just silent guests in our guts. They play an important role in our physiology. They will synthesize important metabolites. They will help us digest our food, and they will participate in the maturation of our immune system. And in the last decades, we've come to realize that the gut microbiota was super important in a number of important diseases, ranging from inflammatory bowel disease and C. difficile infection, but also to diseases that are far away from the gut, like Parkinson's disease or anxiety. And so that gives us really an incentive to understand the gut microbiota better, to understand its role in diseases, and to eventually be able to act on it in a way that might bring health benefit to the host, which is us. And that's really what I'm trying to do. So I'm actually a chemist by training. So there's a chemistry theme here a little bit. And I'm interested in drug discovery. And I think the gut microbiota is really an exciting organ to target in terms of drug discovery. So how can we do this? In other words, how can we go from what would be a disease-associated microbiota to a quote normal microbiota? There's several ways. You can do a fecal microbiota transplant, or FMT, which is quite simply taking bacteria from the stool of a healthy person and transplanting them to a patient. And that, that's an interesting approach. However, it does has issues of standardization and safety, and it's a little bit gross. You can also use probiotic approach, where you introduce live bacteria, but typically this will be one or a few strains of bacteria. And I think you'll understand that if you want to replace a full microbiota, you cannot do this with just one or two strains of bacteria. So that has issues as well. And the approach that we are interested in is the prebiotic approach, which you might not have heard about. The prebiotics are molecules that will feed and support the growth of good bacteria in a way. And this idea of using food for bacteria to support their growth is not something new. Nature uses that trick. In the breast milk, you have human milk oligosaccharides, which are complex sugars. And these sugars are there to sustain the growth of specific bacteria in the developing gut of infants. And so you can do this to go from a disease-associated microbiota to a normal microbiota, but you might also want to modulate what would be considered a healthy microbiota to a different microbiota that has a different phenotype. For instance, you might want to activate the immune system in the context of cancer to help fight cancer, or you might want to dampen the immune system in a way that will help in inflammatory bowel disease, for instance. And all of the approaches that I mentioned before can still be used for this. And it's also interesting to mention that drugs can do this sometimes in a way that might be desired or not necessarily desired. So that's an interesting thing to think about. So if we come back to prebiotics, we think we can use prebiotics to modulate the composition of the gut microbiota. So what are prebiotics? Well, in our food, in fruits, vegetables, and grains, you have these complex chains of sugars, complex carbohydrates. Think of dietary fibers, which are really important for a microbiota. And the reason is that these complex chains of sugars are too complex for our digestive system. We simply do not have the enzymes to process them. Whereas a microbiota, with its diversity, encodes for the thousands of enzymes that are necessary to break apart these complex sugars into simple sugars that the bacteria can metabolize. So basically, these molecules will go through our digestive tract undigested. They will reach the large intestine, and there they will feed gut bacteria. The problem is that we don't really know very well enough who in the microbiota is eating what exactly. And this is what we're trying to answer. And one of the techniques that we use for this is that we've attached the fluorescent molecules on the complex glycans. We feed that to microbiota samples, and the bacteria that will eat the sugar, the complex glycan, will become fluorescent. And then we can use as fluorescents to pick them apart and identify them and cultivate them and study them. And this is one of the techniques that we do in the lab. And the idea is that we might be able to use this eventually to treat diseases like cancer, which is one that we are interested in the first place, and C. difficile infection, and other important diseases. And so I just want to thank the people in my lab and our multiple collaborators on this, because this is really a multidisciplinary science. So I will be happy to answer any questions in the breakout room. Thank you.